Yeah? Yeah? Okay. Hello, everyone. I hope you had a nice lunch and a bit of networking as well. So let's going to start for our second session of the day. So we are now moving on to the next theme entitled New Tools, Technologies and Success in Cell and Gene Therapy. This session will be divided in two parts. The first part will have two speakers from GenScript, Rouba Najjar, Senior Product Manager, and Vivian Zhang, Product Manager. Vivian actually will be not present, but we have recorded her presentation. And the third speaker is Dr. Dina, postdoctoral researcher at the University of Oxford. So each speaker now will have 10 minutes to present, and directly afterwards we're going to have a 5-minute Q&A, so you can be prepared to ask just after to ask directly to them, okay? After I will explain about the second session, so we can go now to the next speaker. So our next speaker is Rouba Najjar. She's Senior Product Manager at GenScript. At GenScript, Rouba Najjar has worked in the biotech industry for 10 years, more than 10 years, and is currently responsible for GenScript's gene editing and molecular biology products and acting as the voice of customer. So let's welcome our first speaker for this part, Rouba Najjar, with the presentation title, Enhancements in Gene Therapy Workflows. I guess will come.
Thank you, Elaine. Hi, everyone. So, as Elaine already mentioned, I'm the product manager for gene editing. So for my presentation today, that's what my focus is on. So I'm joining you from GenScript. We've been around for more than 20 years. And in the last year or so, we've really been focused on enhancing our gene therapy solutions. So from a CRISPR overview, I'm sure everyone probably is aware, if not an expert already, on CRISPR-Cas9. So this obviously won the Nobel Prize in 2020, and really what I have it up here is just a reminder of the way it works. So basically you have your guide RNA that attaches to your CRISPR protein, typically a Cas9 protein, and it takes it to a specific site in the host, and it enables the Cas9 to go ahead and clip that specific site. So it enables basically a knock-in, a knock-out, or potentially other things that you could be working on, such as base editing. So some of the advances in the applications over the past couple of years, of course, genome engineering, specifically therapies, cell and gene therapies, but also very interesting, there's been a lot of advances in industrial biological products, such as crops and biofuels. So CRISPR has really revolutionized some of those industries. And in my opinion, some of the more interesting things that CRISPR has been able to do is more of the detection. So, for example, the detection for more of the viral infectious reagents. So I'm sure everyone has heard of Sherlock, detector, or inspector, and those are all acronyms for some of the kits or some of the solutions that are actually very patented right now, that are basically taking specific locators in the sequence of a virus, looking for those genomes and detecting whether or not there's an infection in the host. So it's a really interesting way that has leveraged CRISPR. One of the new advances for that was actually COVID detection kits. And also some of the detection kits have been focused on agricultural and food safety. So some of the impacting factors and technological challenges, so outside of what we already heard from some of the speakers were, of course, for CRISPRs were the cost, some of the compliances, some of the logistics, and of course the IP landscape. There's a lot of complexities around every single aspect of those. But for my portion, I was actually more focused on the impacting factors and technological challenges. So when you're working with CRISPR, there's a couple things that you want to keep in mind. One is the guide RNA. If you don't have a good variety of guide RNA where your DNA sequence, you're not taking into account some of the challenges of the nuclease that you're working with. So With that said, you just need to make sure that you're working with a good guide RNA. So there's, of course, the two segments, and depending on what kind of Cas nuclease you're using, you can potentially just be working with a CRISPR RNA or both, the trans-activating CRISPR RNA and the CRISPR RNA. Of course, the Cas nuclease itself is going to be a challenge. There's different nucleases. I'm sure you all have heard of a lot of the newer advances. So Cas9 is really well known, but there's new ones such as phage, such as Cas12, Cas13, a lot of the different nucleases that target different sites. So for example, Cas12, more of the recent one, it has a completely different PAM sequence. So it enables a wider sequence. So for example, if you're working with AT-rich regions versus a CG-rich region. And the different Cas nucleases also impact the way that you're doing your cleavage. There's a lot of delivery mechanism challenges as well. So it's really interesting to hear some of the previous speakers speaking about more of the viral delivery mechanisms. It seems that there's a lot of advances around that as well, a lot of focus on RNP delivery as of late. And then the last thing that is a huge technical challenge for gene therapy is the cell type that you're using. So if you are working with viruses or plasmids, you're relying on the cell to do the transcription and the translation. And of course, that also means that by the time you infect your host with the vector or the plasmid, you're having a lag between when it actually gets infected and when actually the therapy takes place. So also another thing to keep in mind is that when you're working with different cell contexts, the actual cell might have different effects on what kind of delivery mechanism you end up using. So let's say you're working with a virus and you're working with a specific cell type. That virus might not attach the same versus a different cell type. So for my portion, I actually wanted to really emphasize the RNP complex. And the reason why is because there's actually a lot of work that has been done in the last year that speaks of the benefits of RNP complex. So RNP complex is when you're taking the guide RNA, you're complexing it with the nuclease, and you're delivering that protein as a whole into the host. So sometimes you're encapsulating it with a lipid nanoparticle. Sometimes you're delivering it actually as a naked form. So there's a lot of benefits to that. And again, I just mentioned it a little bit earlier, where you're delivering it and right away you're performing your gene editing. You're not relying on the cell to actually transcript and translate the guide RNA and the protein itself. Also, another thing that to keep in mind is because you're not working with a virus or a plasmid, you're actually reducing some of the challenges that you see with those systems. So that includes mutagenesis, toxicity, and some immune responses. So I heard some of the speakers earlier talk about the challenges with that, and of course there's definitely concerns with every platform that you're working with. So to keep things simple, I'll go into some of the things that we have done. So we've developed a couple different nucleases that, of course, are well known. So Cas9, Cas12, and 13. And again, those are more specific to what kind of target nucleic acid you're working with, but also the site that you're working with. So for example, 9 versus 12, they have a couple different sequence-rich regions that you want to target. Also, we've really focused our efforts on developing solutions that are more targeted to GMP compliance. So we now have a product line that is the Cas Ultra. And you see that we have 3 different levels, 3 different grades. So we have the basic, the GMP grade, and the RUO, which is the research. And so basically what you have is a nuclease for every step of your workflow. And I'm going to highlight some of the data that we have very, very briefly, just for the sake of time. Basically, for knockout efficiencies and knock-in, you see data in front of you. Again, for the sake of time, I'm going to focus on this particular product, which has tenfold enhanced specificity when you compare it to the wild type Cas9. And what I want to highlight is that when we compared it to competitors, what you see is that there is a pretty high knockout efficiency in cell lines. But when you compare it to competitor 1 and 2, there's a slight reduction in the efficiency of our Cas nuclease. And that actually is explained by the low off-target effects. So even though it's slightly less with efficiency, it actually is a lot higher with the on-target effects. So with that said, what you see in front of you is actually the off-target effects by percentage. So the Ultra ASP Cas9 versus the competition, there was no off-target effects that we were able to detect using OT1 and OT2 sites versus the competition, which you can see is, especially when you get to the 7.5 picomolar, is approximately 20 or 30%. So basically a significant change in the off-target effects. I'm gonna basically, this is the knock-in as well, and you can also see the same story here, where basically payload for the ESP Cas9 was significantly enhanced as well. These are some of the quality control specs, and I'm gonna stop now because I'm being told that time is up. Any questions? Thanks.
Hello. So, are there any questions?
Yes, I'm wondering what assays you use or analytical methods are used to evaluate unintended, not only off-target mutations, but on-target mutations as well in your pool of cells that you're analyzing. Are you using long-read DNA sequencing, for example, to ensure that you actually capture all of the outcomes, both off-target and on-target?
That's a great question. I actually need to get back with you on that. I don't know exactly if they did a long read sequence, so I'll have to get back with you on that.
Anybody else? Okay, so let's move on to the next.